Hello, so I'm Keegan Burrow and I'm presenting some data which is an extension of the work that I presented at the last sheep milk conference. This time though, rather than focusing on the effects of sheep milk consumption on the mineral composition of animals, we're looking at the effects of sheep milk on bone structure and development. So, first of all, I'm just going to give you a bit of background because until I started this research, I wasn't an expert on bone, so I expect probably none of you here are either. And then I'll go through some of the methods we've applied and go through two of the types of analysis that we've used during this study. So, just to have a bit of background, because I think it's really important to bear in mind that bone as an organ system is really, really complex, but also really, really interesting and exciting. It doesn't just provide things such as the ability for people to walk and run, but bone also acts to protect organs, it acts to produce blood cells, not only red blood cells, but immunological cells, and it also acts as a key mineral storage vessel throughout the body. Now, in order to achieve this, bone is made up of a number of different phases, and the important part for my work is particularly the mineral phase, but I don't want to just focus on that. I want to also highlight the fact that bone interplays and has an organic phase associated with it as well. This allows for the fact that bone develops and grows right throughout life, and it also never stops changing. And that brings me to sort of the next point. And obviously in the first session, there was a lot of discussion around ageing populations and around issues such as osteoporosis. But something to bear in mind is for good health and long-term health, we've also got to think about the development during particularly young ages. And so issues such as rickets, though not very common within New Zealand, can have issues associated with long-term health effects and issues associated with bone and skeletal development at a young age can lead to problems later on in life. Okay, so that's probably enough about bone because I'm going to sort of go through the results in relation to that in a bit more detail. But what's really important to note, and this is sort of where the opportunity for this research comes into play, is the fact that though milk and dairy products have been shown to help in terms of mineral absorption, such as what I showed at the last conference, and have been shown to work in terms of bone development and bone health, you know, everyone says make sure you drink your milk to grow up and have healthy, strong bones. Very little of the empirical work has been associated with sheep's milk, and there's a big hole in the literature associated with that. Now, I probably don't need to tell you all that sheep's milk is really unique and is really different than other milk types. That's why we're all actually here today anyway. But I do want to highlight the fact that particular fractions of sheep milk are really important and really interesting in relation to bone health. That is the lipid fraction, the mineral fraction and the vitamin fraction. And the fact that sheep milk contains higher levels of all of these aspects, particularly compared to cow's milk, the dominant milk source, but also in comparison to goat's milk to some extent, is really, really important. Okay, but enough about this boring background, and let's get into the work that I've actually been carrying out, which is to the aim of our work was to look at the impact of the intake of sheep milk compared to cow milk in a growing rat model. We wanted to look at the structural properties of these bones, and we also wanted to have a look at the physical properties of bones. So what do the bones look like on the inside, and then how easy is it to break them? In relation to this, we've applied a couple of key techniques, and these have been carried out down at the University of Otago using micro-CT analysis and 3-point bending tests. Now, I'm not going to go into too much information around how the samples within the study were obtained because we bored you to death with this information last year at the conference. But what I am going to highlight is that we're working with bones that have come from an animal feeding trial where newly weaned male rats were fed whole sheep's milk, whole cow's milk, or sheep's milk diluted to have the same total solids content as cow's milk for a period of about 36 days, and then the animals were euthanized, and we've harvested various organs, but also the bone material to carry out the testing. In relation to the bone, we were able to test a range of different variables with this material through both destructive and non-destructive testing. So there's the micro-CT testing, then break force testing, which is a semi-destructive technique, and then we also have mineral analysis by ICP-MS, which I'm not going to talk about today because I already have far too much data to get through. Okay, now let's move on to actually some of the interesting stuff, which was the techniques that I've applied throughout this research and the data we have got. So I've bandied around this terminology of micro-CT analysis, but it's probably going to be a good idea to explain what this is. Micro-CT analysis, or X-ray computer topography analysis, and scientists have awful acronyms for everything, I apologise in advance, is effectively a really, really high-powered X-ray tool. So if you go to the hospital and you have a broken arm, you get your arm x-rayed and they can see what's happening with that bone. What we're doing here is basically the same thing with the samples that we've collected, except in a much higher detail in 3 dimensions. This means that we can get a lot of information out of the way that these bones are structured internally and see any effects of the different consumptions of the mix. Now, in relation to those internal structures, there is a couple of different types of internal structures we did assess. So, bone has got areas of cortical bone and tubercular bone, and basically, to sum it up, cortical bone is your long bones, the hard sort of classical bone structures, and tubercular bone are sort of more like a sponge-like structure that's present on the inside of bones. But instead of just describing it, I'll show you some of the images that we got out of our study. So these are image slices that have been taken from the 3D reconstruction of the bones that we have analysed. At the top you have the cortical bone, which is very obviously the sort of the deep, it's black area there, and on the bottom we have the tubercular bone, which has sort of got a more flaky area, and all of the black, it basically represents the mineral phase of these bone samples. But as you can probably tell, it's really, really hard to actually tell what the hell is happening from these images. And so we ran all of the images and the data we obtained through some image analysis software. I'm not going to bore you with the details because it basically was me swearing at a computer for months trying to get it to work. But from that, we were able to measure a range of different variables and characteristics in these bones and in the microstructure. I'm only going to give you a couple of examples from each type of bone. So here we have bone volume, which is basically the total amount of the cortical bone that is present. And then we have bone surface density, which is basically how tightly packed in we have that bone. And what we can see with this data is that there is a pattern occurring going from cow's milk to diluted sheep's milk to sheep's milk, where we're seeing an increase in these positive bone parameters. Likewise, we're seeing the same sort of effect occurring in the tubercular bone data. So we've got different measurements again, and these are just a selection of the different variables that I've pulled out. So we have bone surface to volume ratio and tubercular number. Effectively, once again, how much bone is present in that space, and then how much of that relates to sort of the spongy structure. We're seeing the same pattern or trend occurring in this data as well. But sadly, we found that none of these results were statistically significant individually. But thanks to some help, particularly from ag research, we're able to look at this data in a slightly different way. And this was using multivariate analysis, which only makes sense because bones aren't just a bunch of individual parameters, but as a whole system. So each bone, all of the different ways in which the structure form, all play into how this bone actually is going to function in the long term. And what we found was that there was a significant difference in the bone characteristics between the rats that were fed sheep's milk and those who were fed cow's milk. And if you think back to those patterns that I pointed out, we're seeing an increase in the positive parameters in the animals that were fed the sheep's milk samples. And that's sort of the take-home point from this part of the research. Moving on to sort of the more fun part of the analysis was basically me going into a lab and breaking things. So this 3-point bending test is something that if you've done any engineering, you may have an understanding of, but I'm not going to bore you too much. Effectively, we put the bones into this really fancy breaking machine, and we were able to measure a pile of different parameters associated with the changes. This here is just a close-up image of how the bones were positioned with the machine, and it was just a standard methodology. This machine then just measures the amount of force and the amount of deformation present, and then we can extrapolate a lot of information out of that data. But sadly, none of the results that we obtained through this information were statistically significant. So we weren't seeing any differences in the mechanical properties of these bones due to the treatment. And once again, this is just a selection of the various parameters we have measured. But that's not necessarily the end of the world, because I want to highlight a couple of things around these bending tests and breaking tests. First of all, it's really common for mechanical testing and structural testing not to always line up, and work on cow's milk and goat's milk and different fractions from that milk have shown this as well. But furthermore, I think it's kind of important to think of the fact that cow's milk is already seen as such a good contribution to the diet. And what we're seeing here is the fact is that though sheep's milk is improving the structure of these bones, we've already got the maximum mechanical characteristics we can with that diet of cow's milk. So adding the sheep's milk is not necessarily improving that in a way we can analyse. Even though I do want you to take home the fact that we're starting to see some advantages for sheep's milk in comparison to cow milk on the structure of bone. And what's really important to consider for this isn't a case where these animals were fed far more than their dietary requirements for various minerals, lipids and fats. They were provided with a basal diet and then provided these milks on top. So to be able to see these results and see some differences is quite a good indication for the future. And though we're not seeing any differences in the mechanical properties, this is only looking at a relatively short term. And this links back to when I was saying about the fact that nutrition at young life is going to affect nutrition at an older age. In this case, we're only looking at the way that the mechanical properties of these bones are occurring while these animals are still quite young. And so into the long term, and as this extends longer, there may be potential for benefits to accrue. And we're currently working on some planning to carry out some further studies to look at the potential of milk in a mineral deficient diet to help sort of cover some of the problems that I've just highlighted with my own study. In terms of acknowledgements, there's lots of individuals from the University of Otago I have to thank, particularly in the Department of Anatomy and the Confocal Microscopy Unit. Obviously, all of the various sponsors in terms of the conference. And also I'd like to thank Agmart as well, because they're assisting me with being able to carry out our next animal trial, which is going to build on this work, and hopefully I can come back next year at the conference and present some even more new and exciting data for you all. Thank you.